Hey guys, welcome to our YouTube channel, and today's video is going to be about karyotyping. And before we go into karyotyping, let's talk about what cytogenetics is. So cytogenetics is basically how we study these chromosomes and identify certain diseases related to it. The way we identify these diseases when we look at the changes in number of chromosomes and its structure. So karyotyping is basically when we organize certain chromosomes of an individual and we can identify if that individual has any genetic problems or any disorders which is caused due to it. There are three types of chromosomes: metacentric, submetacentric, and ectocentric. When you look at metacentric, it has one centriole and the P arm and the Q arm are equal in length. When you look at submetacentric, the P arm is quite short and the Q arm is long. And when you take ectocentric, the P arm is so short that it might not be visible. The Q arm is quite long. And you can see in all three of these, the position of the centromere is quite different. So the important thing about how mitosis plays a role in karyotyping is we know that there are 4 stages in cell division, that is prophase, metaphase, anaphase, and telophase. So the main reason that we take the metaphase stage is because we know that in that stage the chromosomes are thick and tightly packed and they are also condensed. So which means that in this stage the chromosomes will be very much visible to us compared to all the other stages. When you look at the microscopic view, we are looking at 3 main factors, that is the size of chromatids, banding patterns, and its centromere position. This diagram shows us that in a microscopic view, these are the stained chromosomes. As you can see that it's not really clear. So what we do is we process this image in a computer and we get a much detailed contrasted image which shows us clearly each banding pattern, and we can clearly see location of each centromere as well. This is a karyogram of a male and a female which are normal. In normal male and female, the main difference would be in the 23rd pair. So as you can see that in the male there is one long chromosome which is denoted as X and a small chromosome which is Y. In a female there's 2 X chromosomes. So when you look at the arrangement, you can see that, for example, if we take this chromosome, it has a white tip and bands in the bottom. So this chromosome is very much similar to the first one. Likewise, when you look at this chromosome, by looking at it we can tell that it's similar to the 22nd one. Likewise, when you take this, it's similar to the 21st one. So that's how we arrange it. We look at how each chromosome is similar to each other and we compare it with the reference and we crop it and we attach them, and that's how we form the entire karyogram like I showed in the previous slide. So when you look at the genetic diseases, there are 3 diseases we are going to talk, that is Turner syndrome, Klinefelter syndrome, and Down syndrome. So when you look at Down syndrome, it means that in the 21st pair, the individual has an extra chromosome. The total chromosomes of a normal person would be 46, but in this case, since there's an extra one, it would be 47. As a result, there will be clinical features such as mental retardation and congenital abnormalities. So when you look at Klinefelter syndrome, it's a genetic condition where the patient has an extra X chromosome in the 23rd pair. In this case, it would be X, X, and Y. So the individual might have features such as gynecomastia, osteoporosis, and testicular atrophy. So when you look at Turner syndrome, this affects only females. You can see that in the 23rd chromosome, it's missing a X chromosome. So the individual might have clinical features such as a webbed neck, small size breast, primary amenorrhea, and short stitch.
Thank you guys for watching this video. Hope you liked the video, and if you did, please hit the thumbs up button and also share and subscribe to our channel. Thank you.